So the chapter 9 outline starts on auditory signals run from cochlea to cortex. So the vestibular cochlear nerve, which is the cranial nerve number 8, runs from the cochlea to the brainstem of the auditory nuclei and has the most afferent fibers that are carrying messages from the IHCs. The tuning curve is a graph of the responses of a single auditory nerve fiber or neuron that neuron to sounds that vary in frequency. Each auditory nerve fiber divides into two main branches that it enters the brainstem. And each of these branches then goes to one of two groups of neurons. The first is the cochlear nucleus, which is a brainstem nuclei that receives input from auditory hair cells and sends outputs to the superior olivary complex. And the next one is the ventral cochlear nucleus. The cochlear nuclei takes multiple paths. The first is the superior olivary nuclei, which is a brainstem nuclei that receives inputs from both right and left cochlear nuclei and provide the first binaural analysis of auditory information. It also plays a key role in localizing sounds by comparing two ears. The inferior colliculi is the next path. That's a paired gray matter structure of the dorsal midbrain that receives auditory information. Outputs of the inferior colliculi go to the medial geniculate nuclei, and that's a nuclei in the thalamus that receives inputs from the inferior colliculi and sends outputs to the auditory complex. At the medial geniculate nuclei and auditory complex, neurons are excited by certain frequencies, but they're also inhibited by their neighboring frequencies. So this allows us to discriminate small frequency differences between sounds. Another thing is tonotopic organization, which is a major organizational feature in the auditory system, in which neurons are arranged in an orderly map of stimulus frequency, with cells responsive to higher frequencies located at a distance from those responsive to low frequencies. So this is a 2-deoxyglucose mapping, is an idea of how you can see this. After injection, an animal is exposed to a tone of a particular frequency. The most active neurons will take up the most 2DG. Post-mortem processing shows which cells are most active when stimulus frequency is presented. Primary auditory cortex is the region of the superior temporal cortex in which auditory processing occurs. It's located on the upper surface of the temporal lobes and can also integrate other non-auditory information. So the next major section is pitch information is encoded in two complementary ways. The first is a minimal discriminable frequency difference, which is the smallest change in frequency that can be detected reliably between two tones. For humans, it's about 2 Hz for sounds that are under 2,000 Hz. There are two signals from the cochlea that appear to inform the brain about pitch. The first is place coding, which is the encoding of sound frequency as a function of the location on the basilar membrane that is most stimulated by that pitch or by that sound. There's two parts to this. The first is treble, which is an aspect of pitch corresponding to the subjective experience of high frequency sounds, and that has to do with the activation of receptors near the base of the cochlea. And then there's bass, an aspect of pitch corresponding to the subjective experience of low frequency sound, and that has to do with activation of receptors near the apex of the cochlea. The second is temporal coding, which is the encoding of sound frequency in terms of the number of action potentials per second produced by an auditory nerve. So phase-locked action potentials are at the stimulus, so they occur at a particular point in each sound cycle and directly reflect the number of cycles per second. Higher frequency sounds will have an increase in frequency of action potentials that may encode auditory frequencies as an integer multiple. This allows auditory systems to overcome the maximal neuronal firing rate of about 1,000 action potentials per second, and temporal coding functions up to about 4,000 Hz. So frequency properties of a sound are coded according to the distribution of excitation among cells and according to the temporal pattern of discharge in cells projecting to the auditory cortex. Mammalian species employ a range of frequencies from less than 10 Hz to more than 10,000 Hz. So there's infrasounds, which are very low sounds, and they're in general below the threshold for human hearing at about 200 hertz. Some animals that can hear that are tigers, elephants, and pigeons. And experimentally introduced to concerts increased its emotional effect. Ultrasound is high frequency sounds that are above the threshold for human hearing at about 20,000 hertz, and these can be heard by bats and porpoises. The next major category is brainstem auditory systems are specialized for localizing sound. So the auditory system accomplishes feat of localizing sound by analyzing two kinds of binaural cues that signal location of sound source in a horizontal plane. The first is intensity differences, which are perceived differences in loudness between the two ears, which can be used to localize a sound source. It depends on the species. It can be due to pointing the ears towards the sound because of shadow sound produced by the head. Sound shadow prevents sounds located from one side from reaching both ears with equal loudness. So it's most pronounced for higher frequency sounds. Low frequency sounds can reach around the head more effectively. The next one is latency differences, which are differences between the two ears in the time of arrival of a sound, which can be employed by the nervous system to localize sound sources. They arise because one ear is always a little closer to the off-axis sound. There's onset disparity, which is the difference between the two ears in hearing the beginning of a sound, and ongoing phase disparity, which is a continuous mismatch between the two ears in the arrival of all peaks and troughs, which make up the sound waves. There's duplex theory, which is a theory that we localize sound by combining information about the intensity differences and latency differences between two ears. So at low frequency, there's pretty much no intensity difference, so the latency difference is very important. Birds and mammals receive information from two ears and use bipolar neurons to derive sound locations from the left and right auditory signals. And this makes timing calculations by comparing inputs from two dendrites. The nucleus laminaris is the nucleus of the primary sound localization in birds. There's also coincidence detector, which is a device that senses the co-occurrence of two events. So each binaural neuron functions as a coincidence detector in the nucleus laminaris, and it maps the map is developed more at higher levels, like the tectum, which is equivalent to the mammalian inferior colliculi.
Oh my God.
So superior olivary nucleus is primary sound localization nucleus in the mammalian brain. And there's the lateral superior olive, which processes intensity differences, and the medial superior olive, which processes latency differences. It doesn't appear that they map the auditory space like they do in birds. Sound localization in these areas is encoded by relative activity of the entire left MSO compared to the entire right MSO. So sound from the right would produce more excitation on the left MSO than the right, and bigger differences between left and right MSO the farther the sound source is from the midline. So there's spectral filling, which is an alteration of the amplitude of some but not all frequencies in a sound. Spectral cues provide information about elevation of sound, and binaural and spectral cues converge in the auditory system at the level of the inferior colliculus. So the next part is the auditory cortex processes complex sounds. It was believed that only the auditory cortex is where auditory sensation and discrimination can arise, but cats can still discriminate different tones following surgical removal of the auditory cortex. So it seems the auditory cortex may be very important for detection of more complex biologically relevant sounds. The auditory cortex evolved to process sounds of everyday life. Most cortical auditory neurons habituate rapidly to continuous tones. An ablation of auditory cortex impairs discrimination of temporal patterns of sounds in cats and monkeys. There's two streams of auditory processing found in the cortex. The first is a dorsal stream that involves the parietal lobes concerned with spatial locations of sound, and the second is ventral streams through temporal lobes that may analyze the various components of sounds. During locomotion, motor cortex selectively modifies the responsiveness of the auditory cortex, helping it to focus on specific stimuli. The next part is experience affects auditory perception and the auditory pathways. Experience with sound affects ability for many feats of sound. For example, music is effective in shaping the responses of the auditory cortex. The auditory cortex of trained musicians shows a bigger response to musical sound than does the same cortex in non-musicians. The earlier the training begins, the bigger the difference in auditory cortex in adults. Heschel's gyrus is where music is first processed and more than twice as large in professional musicians than in non-musicians. Amusia is a disorder characterized by the inability to discern tunes accurately. So, auditory cortex that is involved in perceptually analyzing the music interacts directly with the mesolimbic dopamine-based reward circuitry of the brain to attach a reward value to music that is new to us. A congenital amusia tends to be a problem in recognizing pitch and not the timing aspects of music. Brain imaging shows that right inferior frontal gyrus contains less white matter in people with amusia than people with normal music perception. The next section is hearing loss is a major disorder of the nervous system. Hearing loss is decreased sensitivity to sound in various degrees. There's also deafness, which is hearing loss that's so profound that speech perception is lost. Hearing loss affects about 15% of the population. There's three main categories of hearing loss and deafness. The first is a failure of vibrations to reach the cochlea, known as conduction deafness, in which a hearing impairment that is associated with the pathology of the inner ear or middle ear cavities. In some cases, ossicles can become fused together and no longer transmit sound vibrations efficiently. The next is a problem with the cochlear conversion of sound waves into action potentials, known as sensory neural deafness, which is a hearing impairment that originates from the cochlea or auditory nerve lesions. It can be caused by noise exposure, metabolic problems, infections, exposure to toxins, trauma, and hundreds of different hereditary disorders, and this one's usually permanent. In some cases, hair cell structure and function are damaged from birth. There's a gene called GJB2 that encodes for protein connexin 26 that also is involved in the formation of electrical synapses that can cause issues with hearing loss. Ototoxic compounds are things that are toxic to the ears, especially those that are toxic to the middle or inner ear. One of these ototoxic substances are antibiotics. Loud sounds can also damage the hair cells, and this can also cause a disorder called tinnitus, which is a sensation of noises or ringing in the ears. The last type of deafness is dysfunction of the auditory processing systems in the brain. This is known as central deafness, which is a hearing impairment that is related to lesions in auditory pathways or sensors that include sites in the brainstem, thalamus, or cortex. There's different types of auditory or central deafness, including word deafness, which is a specific inability to hear words, although other sounds can be detective. There's also cortical deafness, which is a hearing impairment that is caused by default or defect in the cortex. It involves difficulty both recognizing verbal and nonverbal auditory stimuli. Strokes can interrupt the projection fibers of the medial geniculate nucleus to various auditory cortical regions, and that's also known as deafness, but they do show startle responses, but they don't actually realize that they're hearing a sound.